Scizor is the program. Its job is to locate peaks, and it's this really simple part of sequencing DNA. Scizor program that claims to be better at locating these binding sites. The common software uses max over finding peaks. The Fusion was able to get Scizor functional, but no one else was really able to. Important lesson to know is that you have to know what to put on software. If it takes a staff of 4 to get one program running on one single computer, it's probably not worth it. The microenvironment project, this is background about it. Previous study system at the NYU School of Dentistry showed that there was a particular microbe more common in the mouth, mouths with tooth decay. So if you have a patient with tooth decay, you will find microbes that are more common. The population is greater in a common, in a regular, than in a regular human. Regular. All right. The method used was to just take scrapes out of the mouths and place them on the petri dishes and see what would grow. Microenvironment project. So which microbes are involved in tooth decay? This is what the question was. Experiments of group of kids with tooth decay versus a control group of health kids, no tooth decay. Take samples from both groups and identify differences in micropopulation. Data from 16SR gene. 16SR gene is a ribosomal gene. This is a gene that's very, very common among all living creatures. Every cell needs a ribosome because the ribosome is where the transcription of the DNA can turn into where the protein is. Okay, so this gene is very conserved throughout various species. So this is what we use as a reference to compare how closely, how evolutionary close, the evolutionary closeness of any of the microbes in the mouth. We use a Roche 454 DNA sequencer. This is just another type of sequencing. Identify species with ribosomal database project. This is the RDB classifier. What this does is you can just put the data in. Actually, one of the options is to create a tree. This is the evolutionary tree that shows the closeness of each microbe to the next. So you can see what the group is, how far apart they are evolutionarily. You parse out the output file. Ribosome. This is the ribosome itself. Little dots. Microenvironment project. Taxonomy data. Sorry. This is what our data looks like. So this is the sequences. Those the flow or the headers. So this is a particular file. Results. The results of the project wasn't what we expected to be. They expected Streptococcus mutans was the microbe they expected to be of a prominent, of a higher population concentration in the mouths of tooth decay, but this is not what we found. What we found was that there's no particular difference between the microbes in the mouth with tooth decay and the mouths with healthy. With the mouth with healthy teeth. We used sequencing, we took scrapes, and we actually saw what was actually there. We didn't put it on petri dish, we didn't see what grows, we didn't go through all that process. Because the error with the previous result was when you put stuff on petri dishes, you actually might have an error and see what grows best on petri dishes, not what grows best in the human mouth. Results for this results would only prove that microbes were best for petri. This is another project we did. This is a literature research project. Well, this is a fairly new. In 2005, they did some research on this topic, but ever since then, they haven't really done any research. So when you start off a new project, you should always check and see what people already know. So you don't just repeat the process. You don't have to reinvent the wheel every time. So a good place to go is NCBI and see what already is published. Background information is intron splicing is When the DNA is copied into RNA, the introns, there's two parts to the DNA, there's exons and introns. Every gene ends and starts with an exon. And the intron, or the in-between part, is what's cut out, and the exons are spliced together. So this is the intron, so the two orange boxes would be an exon, and the intron is just cut out. We don't we don't actually know what the intron is used for, but in this process, it's not really used, it's just cut out. So the exons are spliced together, so now that's alternative splicing is also something else that happens commonly. A couple years ago, this was very controversial. They didn't believe that this happened in normal humans, but in fact it does. It happens all the time. You start off with a regular gene. Yeah, start off with a regular gene. This is exon 1, 2, 3, 4, and then the green are introns. And the exons. So you cut out the intron. And so you have alternative splicing. So this is one gene, but the thing about alternative splicing is you don't have to have every exon in the protein that you end up with. So you might have 1, 2, and 4, or you might have 1, 2, and 3, where 4 is cut out, or where 3 is cut out. It happens all the time. Because the cell actually controls this process. So. Tandem chimerism. Tandem means when two objects are arranged back to back, so right behind the other. And chimerism, chimera is from the Greek mythology term. It's used to describe a creature that's made up of two or more creatures. Two separate creatures. Tandem chimerism, so then describes. This is just a name that one scientist made up, so this is not an official term, this is not very well researched. When I was searching this, there's many different names for the same thing, and a lot of people didn't, the results wouldn't match, but they were talking about the same thing if you read the papers. Tandem chimerism describes a phenomenon which occurs when two genes are transcribed and spliced into one mRNA. So when you have chromosomes, there's more than one gene on a single chromosome, and they might be back to back, they might be next to each other. So what happens is that for some reason the cell does not read the stop codon, and it just reads straight through, it's called a read through. So when you're finished, you actually have a messenger RNA that has two complete genes transcribed onto it. So this is what it looks like. So the blue and red are two separate genes. The blue boxes are the exons, which are normally kept, and then the line intron, which is spliced out. So for some reason it skips the end exon and the start exon on the next gene, and it just combines the first exon and the last exon on the second gene. So this is the problem. What we found is this can't be a coincidence, because a lot of patients with leukemia have the same exact conditions, like they have the same problem. So it can't be a coincidence. So this is actually something kind of in the background. This is not. We're still brainstorming experiments. It's still in the background, is there? We're working on it. Looks pretty cool. This is what a screen looks like. This is a server screen. This is the servers that were used, I think, about 5 or 6 years ago. I think in estimating right now. And you can see how big it is. Basically, that's really the size of a cabinet. That's what the servers are. And this is what we use today. Each box is more powerful than the big beast thing you saw. This is the evolution of technology. This is my mentor, Dr. Stuart Brown. This is my co-mentor, Dr. 10. Acknowledgements, Dr. Stuart Brown, Program Supervisor, Dr. Sad, HCS Class. Any other questions? The tandem chimeras? Yes, well, so there's actually two things that happen. One thing is called, it's two separate topics, but they're both happening in cancer patients, cancer patients. One's called translocation. That's actually when a complete chromosome will split at a random location, and also another chromosome will also split at a random location. And the two, so you say you have chromosome 3 and you have chromosome 5. Chromosome 3 and chromosome 5 split in half, and so you have a 3-5 chromosome and you have a 5-3 chromosome. So this is what happens in leukemia patients. And what we see is that this happens a lot. This happens with great frequency around the same location, so it can't be a coincidence. So we want to actually research this more. And right now, I'm actually supposed to be writing, well, in the future, we will be writing software to simulate a translocation. So we can use the simulations to develop better methods of locating translocations. It's just because it's very costly to find translocations. And better detail tandem chimerism, I guess, is just something that happens. What exactly don't you understand about that? Yeah.
Can you go to the side, sir?
Well, this is what the people, she wanted to know if the streptococcus mutans were the reason for tooth decay. And the results showed that no, they aren't. There's no particular, what the school of dentistry, people that went to school of dentistry actually believed was that yes, there is a correlation between tooth decay and tandem chimerism, but what you have to keep in mind is there's correlation doesn't mean causation. There's actually something called ROCH, KCOH. Postulates. These are 4 steps that every scientist has to use to determine if there is a relationship, if there is a cause and effect relationship between a microbe and a certain disease. If you want to explain these postulates, okay, so first you have to start off with, you have to isolate, first you have to identify these microbes. You have to find them in a high frequency in all patients with the disease. Okay, so, and then next you have to isolate and culture these microbes to be able to grow them. And then when you grow them, you have to be able to introduce this to a new host with no sign of the disease, and the host has to develop this disease, the same symptom, the same disease. And again, you have to isolate the microbe and compare the microbe to the original microbe, and if they're the same, in fact, have a microbe that is causing this particular disease. So, yes. I'll just give you a copy.